In our lab this week, we are going to be making phylogenetic trees using the concept of cladistics. So we are going to be building something called a cladogram, which is going to show the phylogenetic relationship between the organisms that we're looking at. So a phylogenetic tree is basically a branching diagram that shows our hypothesis about the evolutionary relationships between a small group of organisms. So phylogenetic trees are often based on shared characteristics. Now these characteristics can either be morphological, so that means physical characteristics that you can observe about the organism, or it can be molecular, based on DNA evidence, DNA sequences, protein sequences. So down at the bottom there are two different ways that these are represented, and they basically mean the same thing. Any of these nodes indicate the most recent common ancestor. So this would mean that there was an organism here. A population of organism diverged into two branches, one that led to species E and one that led to all of these others. Now we can represent it this way as well, showing the common ancestor here, branching off into organism E and then all of these other groups. And then each of these nodes also represents a common ancestor and a branch point that led to those lineages. So phylogenetic trees are basically graphs. All graphs have an axis where one side means one thing and the other axis means another. So both of these are going to show time and divergence. So in this one on the left, time is going to be the vertical. So the common ancestor that was farthest back is going to be towards the bottom. And then the more recent species are going to be towards the top. And then the other axis shows the divergence. Over here we've got time along the vertical axis. So the common ancestor is down here, and then we've got divergence along the bottom. So we base our phylogenetic tree on shared characteristics. These can be one of two different types. Organisms in a particular group, in a small group, in a monophyletic group, are going to have shared ancestral characteristics. These are characteristics that they have in common because they have a common ancestor. Now we're also going to be looking at derived characters. These are characters that have come about since the common ancestor. So the common ancestor and any others that diverged before that are not going to have those traits, but anybody along the same branch should have those shared derived characteristics. So these phylogenetic trees are going to be hypotheses based on whether or not organisms have shared derived characteristics. Now it's a hypothesis because they can change over time depending on what traits you actually use, which characteristics you're looking at, and any new information that comes up. So as DNA evidence or protein evidence comes up, you can also use that to refine the phylogenetic trees. So we're going to be using the process of cladistics to construct our phylogenetic trees. So phylogenetic trees can be constructed in many different ways, but we are going to be using cladistics, which uses the idea of shared derived characteristics in order to identify your branch points and your more closely related species. So in order to use cladistics, we first need basically a table of all the characteristics that we're interested in using. Now in this table, we're basically working in ones and zeros. If the organism is labeled as a 0, it means that that characteristic is absent in the organism. If they're labeled with a 1, it means that that characteristic is present. So after we have our chart of characters, we need to identify an outgroup. So an outgroup is an individual that you know or hypothesize to have shared a common ancestor with all the other species. Now the important thing about the outgroup is that it doesn't share any of the character traits that you are looking at. Now it might share other character traits, but it can't share any of the ones that you are actually using to make your phylogenetic tree. So you know that it's a common ancestor because it shares traits with all of them, but you have to use new traits that have come about, the derived characteristics that have come about after the split with that outgroup, in order to identify the locations of all your other groups on your tree. After you identify the outgroup, everybody else becomes the in-group. These are the species that are being considered and are more closely related to one another than they are to that outgroup ancestor. So here's an example of our character trait table. You can see on one side we have the characteristics that we're looking at, and then on the other side we have the organism. Now I just want to point out that our lab actually shows these the opposite way, so the table is reversed, and they're going to be more complicated than this table here. We can see very easily these guys share all of these share all of these, etc. So your lab is going to be slightly more complicated, but we're really just looking at how many organisms share a particular trait in common. So let's look at how we might use this information to make our phylogenetic tree. So remember, the first thing we have to do is identify our outgroup. So we have to identify the individual that doesn't share any characteristics with anybody else. In this case, that's going to be the lancelet. So let's start our axis. This is going to be our axis on which we are going to build our tree. And let's put in the outgroup. So the outgroup is going to share a common ancestor with everybody else on our tree, but it doesn't have any of the traits that we're looking at. So it does share some traits, just not the ones that we are interested in using for our phylogenetic tree. So the next thing we need to do is look at which character is found most frequently in all the other groups. What characteristic does everybody else share? In this case, it's the vertebral column or the backbone. So all of these organisms except the lancelet have a backbone. So we know that everybody on this tree has to have the backbone. The first individual, though, to have it is the lamprey. So if you look, everybody has it, but the first one, the very first one that it appears in is the lamprey. So he's going to go on our tree first. So here we can see the vertebral column is a trait that everybody after him is going, after the appearance of this trait is going to share. But the lamprey only has that characteristic. It does not have any of the other characteristics that we see here. So let's move on to the next trait. The next trait that's shared by everybody is the hinged jaws. Now, the individual that has both the vertebral column and the hinged jaws are the bass. So he goes next. So there's our bass, and he has hinged jaws along with everybody else. If we continue on, we see our next trait is four limbs. So the frog has the vertebral column, hinged jaws, and four limbs, and all of the other individuals after him also have four limbs. So we're going to put him as the next one on our branching tree. Now let's move on to our next trait, amnion. So this is the amniotic sac. This means that these individuals are born in an egg of some sort. Now the turtle, you probably know, are born in eggs with shells. The mother turtle lays the egg develops outside of the mom. In leopards and other mammals, the egg develops inside, but it's still inside the mother, but it still has an amniotic sac or an amnion. So all of these guys have the amnion characteristic, but the turtle has all four of those previous characteristics. So the turtle is our next characteristic in our amnion. So we can see amnion, everybody after that split has the amnion. Now finally, the only trait that the leopard has that nobody else has is hair. So we're going to put him down here. So we can look at this in several ways. We can look at this and say, okay, everybody after this trait has that trait. Or we can say this trait appeared in a common ancestor that gave rise to both of these groups. You can look at it either way. So this is a nicely constructed tree that shows the relationship. So you can sort of see the change over time. We have this lancelet that actually looks pretty similar to the lamprey. The big difference is the presence of a backbone. And then you see that the lamprey sort of physically resembles the fish. Once we get to the frog, the turtle, the leopard, we begin to look quite a bit different. And we have to look at traits that are not as easily apparent. But remember, this is a hypothesis, so this could change if there's any more evidence. If you have used really good traits, the DNA evidence is probably also going to support it. If it doesn't, you either have an example of convergent evolution or you have used traits that are not good indications of ancestral characteristics.